Now that we know how a muscle is put together, let's go back to the level of a single muscle fiber and take a closer look at that sort of cell. A single muscle fiber, or single skeletal muscle cell, is surrounded by a plasma membrane, like any cell would be, but we call the plasma membrane of a muscle fiber the sarcolemma. So the sarcolemma is the membrane around a muscle fiber. The cytoplasm of a muscle fiber is referred to as the sarcoplasm. The prefix sarc means flesh and is often used to refer to things related to muscles. We're also going to see the prefix myo a lot. Myo means muscle, sarc means flesh, and both of those are very common in terms relating to the muscles. When we take a look at the sarcoplasm of a muscle fiber, we see a number of important features, including a large number of mitochondria. Mitochondria are the organelles that produce ATP for energy, and so we need a lot of mitochondria within the muscle fiber to provide the energy needed for muscle contraction. We also see a lot of a molecule called myoglobin. Myoglobin is a protein that we find inside muscle cells that's important for storing oxygen. So as oxygen comes to muscle fibers, it can be stored attached to myoglobin to provide a good supply of oxygen within the muscle fiber so that it can continue to make a lot of ATP for the muscle to function. Another important molecule that we find in the sarcoplasm of a muscle fiber is glycogen. Recall that glycogen is the main form of stored glucose in animal cells. Having a lot of glycogen in the muscle fiber means the muscle fiber has a good source of energy, a lot of glucose available to produce ATP so that we can continue muscle contractions. While all of those things are important, they're really not the main component of a muscle fiber. The thing that takes up more space inside the muscle fiber than anything else are the myofibrils. Myofibrils are organized bundles of actin and myosin that fill most of the inside of the muscle fiber. And it's these myofibrils, these bundles of actin and myosin, that are responsible for the actual contracting activity of the muscle fiber. In order for these muscle fibers to be able to contract, they require calcium. So a muscle fiber has to have a mechanism to store calcium. And we store calcium inside a muscle fiber in a structure called the sarcoplasmic reticulum. The sarcoplasmic reticulum is a modified type of smooth endoplasmic reticulum that's been modified to do the job of storing calcium. The sarcoplasmic reticulum forms a network that surrounds the myofibrils so that when calcium is released from the sarcoplasmic reticulum, it's right by the myofibrils in order to stimulate them to contract. Another important structure in muscle fibers are the T-tubules. T-tubules are sort of infoldings of the sarcolemma, infoldings of the plasma membrane that reach deep down into the muscle fiber. These are really important because they're going to carry the signal to contract from the outside of the cell deep into the cell so that all of the myofibrils can contract at one time. And we'll see a lot more about how that process works, about how the signal gets transported along the muscle fiber, how it travels down the T-tubules, how that causes the release of calcium, and how that stimulates the contraction of the myofibrils a little bit later. But for now, it's important to know that we do have these T-tubules, these parts of the plasma membrane that reach into the cell in order to carry the signal all the way into the muscle fiber. To really understand how a muscle fiber works, we have to take a closer look at the myofibrils. When we take a close-up look at a myofibril, we can see some of the proteins that make up the myofibrils. Myofibrils are made of bundles of proteins. The proteins that make up the myofibril are called the myofilaments. And there are 3 different types of myofilaments that I want you to know about. The first are the thin filaments. The thin filaments are made of strings of actin molecules. Each actin molecule has what's called an active site. And this active site is where myosin can bind to the actin. So this is important. When a muscle is not actively contracting, those active sites on actin are covered by a long protein called tropomyosin. The tropomyosin sort of lays on the thin filament and covers up the active binding sites on each actin molecule. The bundles of myosin proteins are called the thick filaments. They're called the thick filaments because they are thicker or wider in diameter than the narrow, thin filaments made of actin. These thick filaments are made of myosin proteins. Each myosin protein has a tail that helps bundle it together with other myosin proteins in order to make this thick filament, and it has a head. And so once we bundle all these myosin proteins together into a thick filament, the heads sort of stick out. These myosin heads are important because it's the head of the myosin molecules that can bind to the actin, which is an important step for muscle contraction to be able to occur. The last of the myofilaments I want you to know about are the elastic filaments. The elastic filaments are made of a large protein called titin. Titin proteins are stretchy proteins that are important for helping to hold the myosin thick filaments in place. Unfortunately, a lot of models showing the arrangement of the myofilaments leave out the titin elastic filaments that are important for making sure that everything stays in place. In order for a muscle contraction to be able to be effective, these myofilaments can't just be randomly scattered about the cell. They have to be organized in a specific structure. The specific structure that we organize these myofibrils into is called a sarcomere. The sarcomere represents the specific organization, the way these myofilaments are arranged relative to each other. And the sarcomere is what we call the contractile unit of the muscle. A sarcomere is what actually gets shorter to make the whole muscle get shorter. We put the sarcomeres together to make the myofibrils that fill each muscle fiber. Let's take a closer look at sarcomere structure and how we arrange these proteins in order to get an efficient contraction. I want to start with the structures called the Z discs. The Z discs are lines of proteins that everything else is going to end up being connected to. The Z discs mark the ends of the sarcomere. So one sarcomere goes from Z disc to Z disc. The Z discs of each sarcomere are then connected to the greater structure of the muscle fiber. These Z discs are connected to the sarcolemma of the muscle fiber, so that when the sarcomere contracts, bringing the Z discs closer together, the whole cell is going to be contracting to get shorter. It doesn't do us any good if we just move the myofibrils inside the muscle fiber, but the fiber itself doesn't move. The Z discs connect to the cytoskeleton and to the sarcolemma of the muscle fiber, so that when the sarcomere gets shorter, the whole cell gets shorter. The Z discs also provide the scaffold for holding the other myofibrils in place. Our thin fibers of actin connect directly to the Z discs. So you can see the thin actin filaments all going back and connecting to the Z discs. The elastic fibers, unfortunately not shown in the drawing, also connect to the Z disc. And the elastic fibers are almost like big heavy springs that are suspending the thick filaments in the middle of the sarcomere. It's the careful arrangement of the myofilaments within the sarcomere that gives us the dark, light banding that we see when we're looking at a muscle fiber. The dark areas that we see in this striated banding pattern are called the A bands. The A band shows us where the thick filaments of myosin are. That's why they look darker, because we've got all the myosin filaments in there. Part of the A band has overlapping thin filaments in it as well, but what defines the borders of the A band is the length of the myosin filament. The lighter bands that we see are referred to as the I bands, and that's where we see the thin filaments where they're not overlapped with the thick filament. And running right down the middle of the I band is where we find the Z disc. This line provides a nice overview of all of the layers that we talked about of organization to make a muscle. We start with the myofilaments, the actin and the myosin and titin, that are then arranged into sarcomeres. The sarcomeres are arranged end to end to create a myofibril. The myofibrils, along with the sarcoplasmic reticulum, the sarcoplasm, the mitochondria, the glycogen, the myoglobin, and the sarcolemma, make up a single muscle fiber, a single muscle cell. Each muscle cell is surrounded by the epimysium, and then we bundle muscle fibers together to make a muscle fascicle that's surrounded by perimysium, and then we bundle all of the fascicles together to form a muscle that's covered by the epimysium. Those muscles can be grouped together and held together by connective tissue called the deep fascia, and then finally that's covered by the hypodermis, which is going to connect it to the skin.